Well, as I said earlier, I'm really excited about having Michael Snyder join us this evening. Michael's actually one of the first investigators I met when I came to Stanford 8 or 9 years ago, and we put in a large grant together and working on that proposal, even though it wasn't awarded. It really set the stage for us thinking about things around digital and mobile technologies. And no one is more enthusiastic and committed to us understanding how health, wearable and digital data can really help us understand disease and importantly improve outcomes in the future. Dr. Snyder is a professor of medicine here at Stanford. He's the chair of genetics, and he's the director of the Center for Genomics and Personalized Medicine. He has really pioneered the use of, quote, big data, and big data means a lot to different people, and I hope Michael's going to share with us his perspectives. And he's used that approach along with multiomics to advance scientific discovery and transform healthcare. In fact, his lab, his trainees, his collaborators have invented many technologies that are now widely used in medicine and in research. And his application of omics and wearable technologies to perform longitudinal profiling, similar theme as I was talking about with Project Baseline Health Study, is really helping us identify when people are healthy and when they are transitioning to illness, and to be able to do that earlier. And he's actually made, and I hope he'll share with us tonight, perhaps some really important observations around COVID-19. And many of those observations are now being used around the country, if not around the world, to help us further understand the pandemic as well as other diseases. So, Michael, thanks for joining us this evening, and look forward to your comments and hopefully a lot of discussion from the participants in Project Baseline. I'll hand it off to you.
All right, well, thanks. Let's see, not able to share it now. I can. Great. So it really is a pleasure to be here. I'm gonna just shrink a few things. Move some of the things that are in the way. All right. Well, many of you may not know. Well, first of all, I actually met Ken through Project Baseline. I don't know if he remembers that, but that's, I think, how we first got introduced. And I was actually part of the planning of this early on. And a little backstory there, we had just published our first paper on one person, happens to be me, trying to use this kind of approach, big data, to better understand and manage health. And actually, two people showed up in my office. It turns out it was Vic Begram and Andy Conrad, who said, we want to run this big project that Google's going to put $1 billion into. We're going to line up all kinds of partners. We're going to profile zillions of people. And so I was actually involved in some of the planning of this early on. I wound up kind of dropping out because we spun off a lot of companies, a little worried about conflicts. But anyway, it was real fun to be part of this, meet Ken and work with the team there. I'm not even sure how many, because I can't see all of you, how many are still part of that team, but it really was a lot of fun and we had a great time, and you guys have done amazing things ever since. So what I will tell you about is our labs work that actually is very much in this general area. And I think you'll see where we can go and what we can do with this. This is a research project, not unlike Project Baseline. It might be a little less applied and a little more researchy, but it's really turned out to be very, very powerful in many respects, as you'll see. And I will say at the outset, I'm totally conflicted. We've spun off lots of companies, as I mentioned, many of which are exactly in this space. And so, yeah, we can talk about that if you like. So I'm going to start with the premise that many of you may or may not agree with. I actually think medicine's broken, and is broken in many, many different ways. And some of these ways are illustrated here. It's very focused on treating people when they're ill. As such, it's very reactive. When people are healthy, we typically don't measure very many things, and some of those are of questionable value, I would say. And typically when you're healthy, you're not measured very often. But the biggest problem is this one here. Nearly every decision about your health is based on population-based measurements. I think all of us would agree it's better to be focused on keeping people healthy as such, be very proactive. We're certainly capable of measuring many more things than we currently do, as all of you would know as you're getting measured in this project. And the frequency, I would argue, should depend on what your risk is and what your personal trajectories look like. And then lastly, we would say that, decisions about your health should be based on individual-based measurements, not population-based measurements. Most physicians say we do that, but I would argue it's not at the level it should be. And so we're big believers we should be focused on precision health rather than precision medicine. And that is, in fact, a major theme of the medical school here. So just to illustrate this point, you've been told since you were little that your temperature when you put a thermometer in your mouth is 98.6, and if you actually look at the data out there, there's one typical say, there's a number out there, that number tends to be off. 97.5 is a more typical median number for people's temperature. I myself am 97.3. I've been dropping a little bit over the last 8 years. But the more important point is that there's a spread. This is a 25th quartile, 94.6. This is a 75th quartile, 99.1. And so what that means in today's world is if this is your normal average baseline temperature and you go to a physician, they measure you at 98.6, they'll tell you you're healthy, you're at normal range. What are you doing here? Go home. But you're really up 4 degrees Fahrenheit over your baseline. I guarantee you're not healthy. And that's a big theme of our work. These measurements, the kinds of measurements you guys are getting are actually helping define your baseline. And we think they're very, very powerful then for detecting illness at the individual level. So your health, I'm sure you appreciate, it's influenced by many things. Your genome, certainly one of those things, but activities such as, exercise, food you eat, stress all influence your health. Pathogens and environmental exposures also influence your health. We know this qualitatively, but we're in a world now where we can quantify this. We can actually get measurements. We're actually spending quite a bit of time here. Some things are a lot easier to measure, as you know, from the watches you've been wearing. Food is still clunky to measure, but you can measure it. But equally importantly, you can measure the effects of these things by doing basically deep biochemical and physiological profiles on people. And so it was a number of years ago. It actually is about almost 13 now years ago, we started this project for most people to study. It's the smallest group of people, 109, that we've been profiling for roughly 9.5 years, some a little longer, some a little less. What do I mean by profile? Well, we sequence their genome once, and then we do these deep biochemical measurements on folks, these omics assays, if you will. This is DNA methylome, all your RNA molecules, transcript proteins. These will follow out of your immune cells, peripheral blood monocyte cells. That's being collected, I'm pretty sure, in the study. And then we also measure out of plasma, also the proteome. These are important immune molecules, cytokines, metabolites, lipids. We measure the microbiome. I'm pretty sure you guys are giving microbiome still. We measure gut, nasal, tongue, and skin. That turns out to be quite interesting. We do deep clinical tests, questionnaires as well, stress on food and others. And then we also have some advanced tests. I know some of you are getting these stress echocardiograms. It was done on many individuals, and advanced glucose control measurements, things like oral glucose tolerance tests, insulin resistance tests. We're very interested in this because we're very interested in type 2 diabetes because, disclosure, I'm type 2 diabetic. Is actually predicted from my genome. And so we've developed a strong interest in this. And then we started working with wearables, I think about 8 years ago, before the Apple Watch was out. And I'll spend about half my time on that. So one aspect of this is to collect very, very deep data on people. And the other aspect is to do it longitudinally. So we're measuring people. Just going to close some pictures here so I can see better. Okay, so we're profiling people every 3 months while they're healthy. And then if an adverse event comes along, like a viral infection, we'll typically take 5 to 7 additional samplings. And so we're collecting a lot of data. It's denser than perhaps what you're getting measured on over time, but it is a smaller group of folks. So it's letting us do a deep dive. And the timing was totally arbitrary. We don't know how often you should be profiling people. I have my own thoughts there. And we can talk about that later if you like. So you might say, well, why are we doing all this? Well, this group, more than anyone can probably appreciate it, we're trying to understand what it means to be healthy. How does that change over time? How does that compare between different people? What happens at the earliest times of disease to people's, profiles? And perhaps most importantly, from the health perspective, can these advanced technologies like genome sequencing, all these measurements, can they be powerful for actually measuring your health and being used and incorporated into the healthcare system? And with regards to the last point, what we discovered just in the first 3.5 years of profiling was that nearly half the individuals, 49, had major health discoveries, slightly under, actually more like 47. Some people had 2 discoveries. But the bottom line is we found lots of different things, and 2 important aspects of this are, one is that it covers a wide range of areas: hem-oncology, cardiovascular disease, metabolic, infectious, so on and so forth. Another aspect is that it was, this was all found pre-symptomatically, meaning we discovered these things prior to symptom onset. And no one technology actually was responsible for finding these things. Sometimes it was genome sequencing. In the case, we caught someone with early lymphoma that was picked up by imaging, 2 people with precancers from biochemical measurements, 2 people with serious heart issues. This person here actually was picked up through genome sequencing, again, was healthy. Talking about that in a minute. Someone through wearables and so on and so forth. So again, and often as the combinations of technology, several things together said something's off, and then we would do follow-ups to actually see. So the bottom line is a lot of people did learn something by doing this collection, this ensemble of measurements on people. And the way we like to think about this is the way medicine's practiced today. You're getting basically of a 1,000-piece jigsaw puzzle that you might call your health. We're currently measuring, about 8 to 10. And what this project's trying to do is measure more like 800 of those things. You get a much clearer picture of your health. And that's how we view this thing. So just to run you through a few examples, genome sequencing turned out to be pretty powerful just from the first 70 people. Turns out 13 people learned something pretty important. 12 learned something about their risk just from what's called Mendelian disease. These are single gene mutations that put you high risk for certain diseases. And so we actually, in this cohort of 109 people, 1 person had a BRCA mutation. Actually, that person was adopted. So it was kind of interesting for them to find that out. They don't know if they had a family history, if it's spontaneous. And then 3 people had something that turned out to be pretty darn important. I mentioned this one already. This individual, pretty young person, had a mutation in this gene that puts them at high risk for cardiomyopathy. And it turns out they did a follow-up stress echo, and sure enough, they had a heart defect. Her father actually died in 60s of a heart attack, and aunt had one in her 50s. So we think this is the culprit mutation. And so this person is now actually on medication as a consequence of this. Another individual, also pretty young, had a mutation. This gene puts them at risk for certain cancers. We did a whole body MRI follow-up, and sure enough, they had early thyroid cancer, which was removed. They kept most of their thyroid, didn't need then replacement therapy. So that was a very useful thing to find. Coming into the study, we had 9 individuals that were thought to be type 2 diabetic. Well, one of those 9 individuals actually is an HNF1 alpha mutant, if you will, which is a different kind of diabetes called MODY. And they've been on the suboptimal medication for years, actually switched, and now are better managed because of their genome sequence. And then I'm actually a complicated case, actually was predicted from my complex disease profile to be at risk for type 2 diabetes. And then later I got diabetes. I'll talk about that story in a bit. Someone actually found something about their pharmacogenomics that turned out to be important. So even though it's a minority of people that learned something about their genome sequence, if you're one of these people, it's a pretty big deal. It does influence the follow-up tests and follow-up assays you would get done. And as I say, in several people's case, it was potentially life-saving. Now, many of you may know disease risk is really, this is for these Mendelian diseases, and we do want to look out for them. But most diseases are what's called complex disease, where they're thought to be due to they're thought to be due to many genes of low penetrance, of low effect. So if you're if you're unfortunate enough to collect a lot of these variants that are not so good, you can wind up high risk. And likewise, if you wind up having not many of those, you might be protective even. And so people have now built these polygenic risk scores for trying to predict disease risk. And they work well. Timothy's here at Stanford, he's an expert in this area, Jonathan Pritchard and others. And they actually work well for the top, say, 3%. They worked for me and my diabetes, so I'm at the extreme end. They don't really work for most people, and there's likely reasons for that. One is these polygenic risk scores count on you, count on genetics being linear. They add up all these effects in a linear fashion. And for those of us who are geneticists, we know that actually genetics is not linear, so that actually doesn't work so well. So, and likewise, they ignore rare variants. So we've come up with new methods, and I'm assuming all of you aren't, AI experts or anything, so I wasn't going to get into this detail, but I'll suffice it to say that we came up with some new machine learning methods as new ways of analyzing genomes, and it turns out they're really, really powerful. We went into, it's not listed here, but we went into a disease called abdominal aortic aneurysm, which is some semi-common. It's found that 10% of people in their 60s and above die of this because their aorta explodes, and that's how they discover they're at risk. It's a terrible way to discover you're at risk for a disease, by having your aorta blow up and then die. So 90% die when this happens. So, and Phil Sal's an expert. He'd been studying this here at Stanford for a while. And so we came up, first approach, we have two approaches for looking at complex disease. One was actually, we went and sequenced the genomes of 400 and some people, and every GWAS person, they're called GWAS association people, would tell you that that's ridiculous. You can't sequence that, find anything meaningful. Well, it turns out with this machine learning, it's quite powerful. We can look for pathogenic variants inside the coding regions of genes, and then doing machine learning, we actually came up, well, there were 3 genes sort of thought to be involved time. We came out with 60 genes. They're in all the right pathways. It made a lot of sense. We even set up predictive scores. We can actually predict from a genome sequence your risk for AAA. It's pretty cool, actually. And we can also relate it to the amount of HDL you have. So it's actually actionable information. And then more recently, we've come up with 2 other ways of finding the genetic basis of, this one's for ALS, and this one is for COVID severity. So not old folks who get COVID, their immune system's dampened, so they can die or wind up hospitalized. But people are 60 and younger. In fact, even people in their 20s will get COVID and die, and they're thought to be at higher genetic risk. And so these new machine learning methods, again, I'm assuming you're not experts, so I wasn't going to get into too much, but we can map out the regulatory regions and the relevant cell types for this case. And there were 7 known genes at the time, and with this new machine learning method, we came up with 690 genes. It's really quite powerful and explain much more of the heritability of ALS. And in this case, there were 17 and later 47 genes, and using the exact same data set, we came up with over 1,000 genes involved in COVID severity. And we actually won't get into using single cell data. We could actually say, wow, turns out it's a natural killer in T cells that mostly put these folks at risk. So it defines the cell types that are involved in the disease, and then also helps. We're not quite there with predictive models yet. We're working. We can get prognostic values for ALS, like whether you're going to wind up severe or not, but we're not as good yet for prediction. We're working on that. So the bottom line is we have new ways of analyzing genomes for a complex disease that I think is going to be very, very powerful to apply to the baseline data set. So we'd like to do that. This is by Sai Zong, very talented postdoc. All right, so genomics was one part of the equation for this. Other things have turned out to be pretty revealing as well. Imaging. Be curious to see how many health-related things were found through the profiling of the baseline cohort. But anyway, this is a person with early lymphoma that was picked up. They also had elevated blood markers. I mentioned we had 2 people with precancers. This is one of them, young guy, actually. His 2 samples had way more IgM than anybody else's by a lot. And follow-up said he had too many of these producing cells, puts high risk for aggressive cancer. So this is very useful things to find. We also had, through scanning, 6 people had plaques in their arteries. I'm one of them. It's not me, but mine kind of looks like this, where, they're kind of modest, and the recommendation is to increase your statins And I did that. I actually went on PCSK9 inhibitors and because I have a high calcium score, it dramatically drove down my plaque. So we think this, and my cholesterol. So we think this is, again, pretty powerful information. So we were very interested in type 2 diabetes because it turns out I discovered my diabetes through this. So it was originally predicted from my genome sequence. So coming into the study, 9 individuals I mentioned were thought to be at risk for diabetes. Well, 2 people were diabetic who didn't know it. And it turns out there are a whole bunch of people who are pre-diabetic. That's this group who didn't know it. And we wanted to see how do people become diabetic over time? Do they just gradually get there? Does something trigger it? What's going on? And so what we found in the first 3.5 years, and we now have more data, we're analyzing that now because we're running study 9.5 years. But during this time, during the first 3.5 years, 9 other individuals became officially classified as diabetic, and then others had diabetic range values. So the numbers were kind of high, and others became pre-diabetic. So again, the question is, how do you become diabetic? Is it gradual? Does something trigger it? Well, of the 9 people, 7 gradually got there, and 2 got there by gaining weight. So kind of the classic way you think about people getting diabetic, by becoming overweight. And 2 did do that, and their microbiome diversity dropped, et cetera, which you would expect. But others actually didn't gain weight and just gradually got there. So in orange is your fasting glucose, should be around 90. And this is hemoglobin A1C. You like to have it below 5.7 to be normal. 5.7 to 6.5 is diabetic. So this person gradually went up for both fasting glucose and for hemoglobin A1C, and they got treated here. This individual got there through oral glucose tolerance test, which is a different type of assay. And it turns out these tease out different things, but they gradually went up, we believe. In contrast, 2 other people had things stimulate this. This actually is me. This is my data. Here's, again, glucose. Here's hemoglobin A1C. This is 8 years of data. And like I said, we now have even a lot more. But the bottom line is I actually first became diabetic after a nasty viral infection. In fact, I was going in for an insulin resistance test, and I was measured at very, very high glucose levels. And that's a week later, very high hemoglobin A1C levels, basically classified as diabetic at that point. And so in my case, I was able to switch my lifestyle, at least initially. I had been biking, I doubled my biking, started running, and I gradually brought my levels down to what was pretty normal values. And they were running so boringly low, there's like 3 years of data that, I stopped looking at them. It's like a perfect control. Somebody else looked at it here and said, Mike, you realize you're back to your hemoglobin A1C 7.0, you're back to diabetic again. I said, oh, I had no idea. We went back and looked, and sure enough, I'd spiked up again. And guess what? I'd stopped running here. I also got a second viral infection. And so it's not clear which of these trigger. I think it's probably a combination of both. But the bottom line is that, I definitely have a glucose dysregulation that was predicted from a genome sequence. Initially, it was actionable, and it seems to be a case of genes versus plus environment. So our interpretation is I'm genetically predisposed, and then the viral infection triggered it. And this is now what people are finding. You may know this for COVID. 4% of people get COVID wind up diabetic. Most are thought to be type 1s, but some are definitely type 2s. And so, it turns out, you'll see in a minute, I'm pretty strange diabetic. I'm not overweight or anything like that. All right. So anyway, when this came back the second time, I had stopped running. So I started running again. I got it down. I never went all the way down to baseline. It, kind of got 5.7s in the hemoglobin A1C and spike occasionally. It was gradually creeping up. And I was running basically about 4 times a week, 4 to 5 miles a run. It's about as much as my schedule could handle. And it just kept creeping up. So I switched from running to weightlifting, and that did work. I gained 10 pounds of muscle mass. The idea is that muscle is better for glucose homeostasis. Well, that failed. I just kept creeping up anyway. So finally, I bit the bullet as I was getting to hemoglobin A1C around, right borderline around 6.5-ish. And I took metformin, which is the frontline drug. And guess what? I'm a metformin resistant, non-responders, sorry. So I'm a non-responder. So I kept creeping up. I went all the way to 7.5. And so how did I solve my problem? I didn't fully solve it, first of all, but I did get more data. And it turns out, oops, I guess I don't want that picture loaded. Turns out I am insulin sensitive, meaning my cells do respond and also make insulin just fine. So what's wrong with me? Well, I don't actually release it from the pancreas. So my cells will respond, I make insulin, but if it doesn't release from the pancreas, it's very slow to do that. And so it turns out there's a drug for that. It's called rapinolide. And I took that and actually I'm a pretty good responder. These days, I'm on an SGLT2 inhibitor. That works pretty well for me too. So this is what we call precision diabetes. By better knowing what's wrong with me, I'm able to better manage my glucose, get the right drug for the right condition. And so we think this could be done for basically everybody. Okay. All right. Now, one of the most important things we learned by profiling people is that everybody has personal baselines and they're actually pretty stable, believe it or not. So it's just showing 12 people with 10 or more healthy visits. And every color is a different person and every dot's a different visit. And it doesn't matter which ohm you use, transcriptome, metabolome, proteome, here's clinical lab, cytokines. People's profiles cluster according to themselves, meaning you're most similar, your sample's most similar to your next sample. And for me, I have 6 years of data here. You see, I'm the light blue one. All my dots cluster together on each of the ohms. Here's the red person's cluster together, this sort of bluish, there's purple, so on. So everybody has these personal baselines, and they're very, very stable. And not only that, if you undergo a perturbation, say a viral infection, where here's a case where we had people running to their VO2 max, you will shift, in the case of this one, where people ran to their VO2 max, you shift a lot of your molecules, half of them change, but you still look more like your profile, than anyone else. Now, this is a big deal, we think, because this is why you need these individual profiles. So if you want to tell a disease you, say a viral you from a healthy you, the best way to tell is by comparing your suspected disease sample with a healthy one, with your own healthy one. If you try and compare with 5 other healthy people, that doesn't work so well because the difference between people is bigger than the difference between a healthy and a disease state. That's a really, really important concept. The easiest way to tell if you're ill is compare your sample to your own healthy baseline sample. And that you can't miss, very, very easy to spot. But if I try and tell if you're ill by comparing a sample I draw now with 500 other people, that's really, really hard. And so we think it's really important to get your healthy baselines. And that's what all of you guys are doing, right? You're all part of the study. So this is great. All right. So I mentioned that, we have these profiles, they're pretty stable, they're pretty robust. Well, there are some molecules that change and turns out there's seasonal changes and there's changing with age. And so one of the things we realized early on, I think this would be fun to do with the baseline data as well, is that, we could ask a fairly naive question. You've been told since you were little that there are 4 seasons, winter, spring, summer, fall, and they're 3 months each. And if you think about it, who says there's 4 seasons? Maybe there's 15, maybe there's 2. And why would they be 3 months each? That doesn't even make sense, right? They should follow whatever biology is going on. But we were in a position to actually ask how many biological seasons are there, because how many patterns are there in your molecules? So we had been collecting samples throughout the years, over the years, for a number of people. And what we found is that most, the tens of thousands of molecules and microbes, most of them are pretty stable, just like I mentioned before. But there's 1,192 that will show a seasonal pattern. And here's hemoglobin A1C, peaks here, goes down, some others. Some of these are known, most are actually brand new, not known before. Okay, and here's some microbes. So we can say, all right, we have all these patterned molecules. How many, patterns do they group into? And the answer is 2, okay? I should say the participants are all from Northern California, some from Southern California. And you say, and we can show mathematically it's 2. And you say, oh, well, that makes sense. This is California. There should be 2. One should be, winter and the other should be summer. And you would be half right. Here's the winter pattern. These are molecules and microbes that peak in winter. And these are ones that, again, they're high in January, they go down, come back up. And so it has all the things you would expect. Acne, that's known, believe it or not, and a lot of viral infection associated things. People get ill during the winter. The other time, though, isn't summer. It's actually late spring, early summer. It's April, May, which is, in hindsight, what you would expect. It has allergies and asthma. Related molecules are associated here. But there are other pretty important things, too, and I think Ken might appreciate this. Cardiovascular disease, metabolic disease, type 2 diabetics, markers associated with these things are actually peaking in late April, early May. And our interpretation of this is that even in California, people are less active in winter. You know, it rains, it gets cold, like 55 degrees. That's cold for us. We're wimps. And so we exercise less. That's our interpretation. And so a lot of these molecules build up like glucose. And then, you know, spring comes and people are more active and they go back down. There are other things I don't fully understand. Molecules associated with seizures and sleep actually peak around then too. And I don't understand that yet. We think this information is important because there's a lot of clinically actionable molecules as part of this. I won't have time to get into this, but you know, creatinine and others are part of these peaking molecules. And so you could actually incorporate this into your thinking as you're delivering healthcare, because what you don't want to do is that people are on the edge and, you know, winter is coming and they're going to build up some bad markers. Maybe that's a good kick in the butt to get them moving more over winter. Try a little extra effort and things like that. So there are things you could do. All right, the other time when people do actually shifts and some of the molecules that shift is over time, which we interpret as aging. And it turns out we had 43 people with enough information to see how they are aging, if you will. They're basically, we think 5 measurements within 2 years is enough to see how you're aging. And it turns out everybody's aging differently. This one's me, there's 4 years of data. I have much more now, but my coagulation, metabolic, other pathways are going up over time. This person, though, if you look at their top pathway that's changed, it's their cardiac hypertrophic signaling pathway that's shifting with time. And so this person's a cardio ager. I'm again, pretty typical age. This person's more of a heart ager. We later learned this person's stage 2 hypertensive, which sort of makes sense in with pathway going up. We could group, once again, all these patterns into 4 major patterns. I know there are more than 4, but there are 4 patterns of aging. We call them agetypes. So kidney, liver, metabolic, and immune agers. And so every column is a different person. Every row is an agetype. And so the people over here, these 4 people, they're aging in all 4 categories. This person's aging in 3 of the 4. Not much of a kidney ager, but the others are aging. This person's kidney ager, kidney and metabolic. I'm somewhere in here. I'm kidney, liver, and metabolic. Not much of an immune ager. I'm trying to remember which one's it. Might be that one. So anyway, I'm glad my immune system's holding up pretty well. So bottom line is we can actually classify people. And once again, it's clinically actionable. There are clinical markers associated with these agetypes. I'll walk you through the one that's easiest to interpret, hemoglobin A1C. Here, this is population as a whole is going up. That's known in general. But if you look at specific individuals, the dark lines means they're statistically significant. So yeah, these 4 people are going up in their hemoglobin A1C, but then we had 4 other people that went down. And so of course, then you correlate with lifestyle changes. And once you know it, 2 people lost weight, that's these 2. These 2 people went on diets, this person exercised, and so on. So that's probably how they were able to reverse their metabolic aging. There's other things we don't fully understand. There's creatinine, which is a mark of kidney function. The group as a whole went up. That's known to happen over time. But there were basically 10 individuals that went down. And there, what we discovered is 8 of the 10 were on statins. And so we think there may be some relation there. I don't think it's just loss of muscle mass because, we didn't have any data to suggest that was the case. So we'll have to see what's going on. Maybe statins are useful for that. There's other things as well. Right. The other thing we could do too, which is kind of fun, is we could actually see how people are aging based on some markers from other groups. So Morgan Levine has a marker set for aging she calls phenoage, and it's 9 markers. And sure enough, people are aging at different rates. this is their biological age relative to their chronological age. And so you don't really want to be above the line. That means you're older than your age in years. And below the line's where you want to be and have your slope go down, so you're reversing it. But anyway, I wouldn't worry. We don't have huge amounts of data for this to be significant, but the point is that we do see people aging differently. So one question I get asked a lot, Mike, how are you going to scale this thing? You know, what you did, and it's true, we spent millions of dollars on the study to, you know, see what we can learn. It is a research study. And of course, if you're in Silicon Valley, you spin off a company because they think they're better at scaling than academic labs. And so we formed a company called QBio. I don't know if any of this group has done it. You may not have to since you're in Project Baseline, although I suspect there will be things that we're learning that are interesting. Anyway, it's a medical version. It doesn't do RNA-seq, for example, because no clinician would know how to do that. But we do deep data dives. We also do whole body MRI, which I think some of you are doing. And we think that's powerful. Most physicians, Ken can comment, but most physicians will tell you should not be doing whole body MRIs. We say you should, and the reason is that the goal, people say you shouldn't do it because you're going to have nodules, like women have them in their ovaries, men in their prostate. My point is that's not the point. It's not whether you have nodules. The issue is, do you have any growing nodules? So by doing longitudinal measurements, you can actually pick these things up. And so what we discovered by doing these deep data profiles, same thing, just from the first 100+ people, these are a few examples, caught some early ovarian and prostate cancer, even earlier pancreatic cancer, which is almost never found earlier, cardiovascular disease, so on and so forth. And sometimes it was multiple kinds of measurements that tipped it off, like the imaging and the biochemical markers. Other times, imaging would turn out to be powerful, as did all the other things, including genetics and such. So, but many cases, the longitudinal measurements are super, super powerful. So I'm glad you guys are running this longitudinally, because I think that's going to be the key to see the shifts. What's not clear yet in medicine is how often you should be profiling people. Again, I would argue it depends on your risk factors. Anyway, we're recommending as part of QBio every 6 months as part of a research study. We do every 3. All right. Now, our cohort is a very, very eager bunch of folks, and they like to participate in studies we happen to run in parallel. So we have a bunch of people who are did the exercise study I mentioned earlier, where they ran to the VO2 max and we profiled them. Most recently, we did a study around fiber. You hear fiber is good for you. It is. It sits in your colon and supports your microbiome, and it makes short-chain fatty acids. It boosts your immune system, and breaks down into important nutrients like polyphenols and things like that. And it winds up preventing all kinds of different diseases. So fiber is really awesome. And you may or may not know that women are recommended to have 25 grams per day, men 35, but the average person has somewhere between 10 and 15. We're way off the amount of fiber we should be eating. So we wanted to see if we could actually pin down better the effects of fiber. Let's see how we're doing time-wise, because I definitely want to talk about the wearables. Oh, yeah, we're good. All right. So I'll give you a little bit here. Oh, this talks a little bit about, again, further a little on fiber. So fiber, here's your the equivalent of your digestive system. Stomach feeds into 22 feet of small intestine, feeds into 7 feet of your colon. And your colon is just, fiber basically loads up your colon, and that's where your microbiome is, and little bits in small intestine, very little. Most is in your colon. And it's breaking down the fiber and turning it into goodies that you use. It makes short-chain fatty acids, which boosts your immune system, and so on and so forth. So fiber is really, really important for you. The problem is, you say, all right, fiber is good for you, but that's like saying all animals are the same. And they're not. Fibers come in all kinds of flavors. There are long chain, short chains, hydrophobic, hydrophilic, positive, negatively charged. They're all over the map. As I say, they're very, very heterogeneous. So the question is, so which ones are good for you and which ones aren't? And it's been very, very conflicting in the literature. So we decided to see if we could study the effects of 2 very popular fibers, inulin and arabinoxylan. These are very, very common. This is found in Metamucil. This is found in a lot of stuff. And here's some polyphenols that are on the end of those things. They're both polymers. And so we said, well, let's see if we could just break this down. Are there positive effects to these things? If you read the literature, both have been suggested to be important for lowering cholesterol and also stabilizing and lowering your glucose. And, but then there's other reports that say that's not the case. So we basically decided to test for ourselves. We took each of these fibers and fed them as a supplement to our study participants. We had 18 people, roughly half men, half women, and they would take 10 grams of fiber a day as a supplement, either arabinoxylan or inulin, and then they'll go to 20 the next week and 30 the next week. And then there's a washout period, and they'll switch to the other fiber in a washout period. And then we had a mixture of fibers. Everybody assumes mixtures are the best for you. And so again, it's all done as a supplement study, 10, 20, 30 grams, increasing amounts each week. So we wanted to see the effects of these fibers on people's omics profiles. So we did what we're best at, which is profile these folks at each of these times. Very, very dense sampling through the study. And then same thing, we look at lipids, metabolites, proteins, all the things I mentioned before for IPOP we're doing as they go through this fiber supplement study. And what we discovered is that both fibers change thousands of molecules. So here's arabinoxylan. These are the molecules that go up. You can do this with the systematic method called C-means clustering. You can find all the molecules that go up. We also identify all the molecules that go down. And then we can say, well, what's in there? Well, it turns out lots of interesting things are in these patterns. And one of the first things we discovered that's clinically interesting is that arabinoxylan actually does decrease the amount of cholesterol in the cohort as a whole. So here you go. As you increase arabinoxylan up to 30 grams, you will actually reduce the amount of cholesterol folks have. Pretty good too. And inulin turns out has no effect, in spite of some studies in literature saying it does. This mixture actually did also lower it, but not as well. So we think it may be a linear dose effect or semi-linear. All right, so arabinoxylan did decrease cholesterol. Turns out inulin in general did not have any effect on cholesterol, as I showed here, and it also didn't have any effect on glucose, in contrast to what the literature had, nor did arabinoxylan. So this is the one most useful clinically thing we found. Now, by doing our deep chemical profiling, something got messed up here. We also discovered some bioacids actually wind up changing. And the bottom line is, since that's messed up, I'll jump here. We came up with a whole new method, a whole new hypothesis for how cholesterol gets reduced by arabinoxylan. And we think it's actually involves the gut microbiome, and actually you wind up basically converting cholesterol to these bioacids that get removed. This is very different from the prevailing wisdom, as people would say, for how they thought cholesterol got removed. Yeah, so anyway, it's kind of cool. We came with a new method. One thing we discovered is that your microbiome diversity does drop, even during the mixture, and that surprised me. I thought the mixture would increase this. All right. And one of the nice things is that we can actually see how people's microbiome shifts in relation to fiber. So again, remember, the fiber is feeding your microbiome. So if you have more of some, that should shift it. And what we discovered is that there are certain species, these are very good species, they go up in the presence of inulin. Specifically, they don't change much with arabinoxylan. These change with arabinoxylan, but not inulin. These change with both. So the bottom line is you can actually control your microbial composition with these prebiotics. And we think this is going to be very, very powerful for trying to manage people's health through food. I'm a big believer that food truly is medicine. And now we can come up with, understandings of how this works, so we can better design this at a personal level. And we think it's really, really important to manage people's health at a personal level. And here's one reason why. I told you that the group as a whole went down when people had arabinoxylan. There's a good classic example. It's this one. It might even be me, I'm not sure. But there are some individuals that actually don't respond to arabinoxylan. It's like, huh, that's a surprise. But then it turns out the same person will respond and go down to inulin. And so we think there's specifics about their microbiome that they're not able to respond properly, and therefore they don't reward the benefits. That's the hypothesis we need to test. So I don't think it's only going to be the microbiome. I think other things will play into this as well. But we think this is again, the importance of knowing your personal measurements, your personal response, so that you tailor this to you. I mean, if you're a non-responder, you can take all the Xylan in the world, and you're not going to lower it. So you just have to, match it up. Or you take a probiotic that goes with it to actually repopulate. Okay, whoops, went the wrong way. All right. We'll spend the last part talking about wearables. I may not have given myself as much time. We're very keen on these. We got very, very excited about these when, Fitbits came out and Apple Watch wasn't out yet when we first started on this. And we think they're powerful because they just make so many measurements every day. So the cheapest watches will make something like 100,000 measurements a day. And the expensive ones will make more like 2.5 million measurements on people every day. And as you probably know, if you've been tracking yours, I don't know much about the Google Watch. I was wearing one for a while. So I don't know what the latest one does, but they all measure heart rate variability, and they're pretty good at that. Respiration is so-so. Skin temperature depends on the device. Some of them are really good, some not so good. Blood oxygen and blood pressure, generally they're poor from an absolute value measurement, but it turns out they will, their deltas are fine, meaning you can still use them to see changes, even if their absolute values aren't good. And so we think these are powerful because they're tracking you 24/7, 365 days a year, as long as you keep them charged. And not only that, they're worn by millions and millions of people. So 20%, 50 million people of the US population all wears a smartwatch. So we wanted to see if we could use these as health trackers. Again, I think this is now being appreciated, but when we started this 8 years ago, people were just using them as fitness trackers, figure out their habits, and then throw them in a drawer after 3 months. We realized that these, again, should be powerful health monitors. So the way we use this is to actually define people's baselines, and we do this in a circadian pattern. So this is me, my sleep, heart rate, skin temperature over 24 hours. So obviously I sleep at night, wake during the day, occasional nap, back to sleep at night. Heart rate is low at night, high during the day, back down. Skin temperature, for most people, it's high at night, drops, comes back up. So we can define this for every person. Everybody has a different baseline, resting heart rate, skin temperature, blood oxygen, so on and so forth. We're all different. And that's why you have to measure these so you know what your baseline is. And then the name of the game is to look for shifts from the baseline. And I can tell you one amusing thing was the first thing we discovered, I travel a lot, is that your blood oxygen goes down on airplanes. And most pilots know that. That's known before. Most flight attendants don't know. Most passengers don't know. I'm not sure if the group here knows it. But your blood oxygen goes down on airplanes. That's because they don't pressurize the cabin fully. And what's highly variable and not well reported is that the amount just varies a lot from one person to the next. And we went on to show it correlates with fatigue. That's not been reported. Now, one of the things we discovered, the second thing we discovered is you can pick up infectious disease from a smartwatch. And we got into this because we discovered my Lyme disease, of all things, with a smartwatch and a pulse ox. So the backstory behind this is I was helping my brother put up fences in rural Massachusetts. And then 2 weeks later, I was flying to Norway through Frankfurt. As you can probably tell, I measure myself all the time here. You can see if you look up, I'm wearing 4 smartwatches there. I usually use 8 of these devices every day. And so I always measure my blood oxygen on airplanes, and it normally drops. And on this kind of a plane, this is when I was flying from Frankfurt to Oslo. It dropped, normally it would drop to 96. Well, in this flight, it dropped to 90 median. That was quite a difference. It was very noticeable. And when I landed, it didn't return. I noticed that my heart rate was high. That's shown here at low resolution. I later learned my skin temperature was up. I didn't notice that at the time. So, well, and what happens is all pre-symptomatic. So meaning I saw my blood oxygen dropping, my heart rate goes up. I didn't to know about the skin temperature. And then later off and on, I did get a fever. And so I did go to a physician in Norway. He draws blood and then, says my monocytes are up, says, yep, you got a bacterial infection. I recommend you take penicillin. And I said, no, I should take doxycycline. And it got pretty tense for a few moments, as you might imagine. Most doctors don't like their patients telling them how to treat themselves. Anyway, he did give in, gave me it, cleared it up right away. He'd take it 2 weeks. So when I got back, I got tested. Sure enough, I was Lyme positive. I even had some antigens. This is by an antibody test. And it's a well-controlled experiment. I give him blood before I left. I was negative. So I clearly seroconverted during this time. So the bottom line now of this, so they could pick up when I first got ill with Lyme disease, and I didn't have a bullseye, is all picked up with a smartwatch and a pulse ox. So we then looked at 2 years of data I had, and it turns out I was ill 4 times. The Lyme time, 2 viral diseases, one here, and the 4th time I was ill, but not, I was asymptomatic, and I had this high protein, C-reactive protein, a sign of illness. And sure enough, every single time I was ill, I had high resting heart rate, plotted here as outlying measurements, high skin temperature. Again, this is low resolution. You can't miss the Lyme signals. They're huge. Even these you can't miss. They're way away. Most visits are down, or most days are down here, the bottom. Even these are pretty high. So he wrote an algorithm. It worked well for heart rate, not for skin temperature. We call it change of heart, where you take people's baseline on an hour-by-hour basis, and you look for the shift up. It's a sliding window, if you will, for those who follow the sort of thing. And basically, we're looking for as a shift up in your baseline. And then, this shows the delta plot, if you will, shift. And it worked. This algorithm worked retrospectively. Every single time I got ill, we could pick it up with a smartwatch. And we had 3 people, one of whom got ill twice. And same thing for them. Every single time they got ill, we could pick this up. And it was in advance of symptoms, shown here in green or red for me. So the bottom line is we can pick up asymptomatic, pre-symptomatic cases, both actually, with a simple smartwatch. So as you might imagine, along comes the pandemic. We were improving the system, trying to actually build it to scale. Because what we're trying to do now is actually reach the whole planet. 3.8 billion people on this planet have a smartphone. And if we compare that with a smartwatch, we have a health monitoring system. So we're building out our cloud-based system. And then along comes COVID, of course, bam. And sure enough, we've been through all these waves. We're still running 100,000 cases a day, probably more. People are getting tired of reporting it. So anyway, but the numbers are still quite high. And so if you think about what we do now, well, we do 2 things. One is temperature, which is a 300-year-old concept, is still used. And that isn't so great for COVID because a lot of people don't get a fever. And in fact, I don't know about you, but when they use these infrared devices and you walk up in wintertime, they shine on you. I'll measure 33 degrees centigrade. It's a worthless measurement because there's no way I'm 33. I got to be. And they tell me to go in anyway. So you just take a worthless measurement, wave you in. So obviously we need something better than that. PCR and antigen tests, you don't do them continuously because they're a bit of a pain, and they can cost a bit more. So bottom line is we think wearables are a possible great avenue for this. So I'd love you all to sign up for our study. All right. So when the pandemic hits, what we did was right away, we enrolled 5,300 people. 32 of them were perfect. They had wearing a Fitbit, who we partner with first. They were also wearing a smartwatch while they get ill, and they had a diagnosis day and a symptom day. And what we discovered, first thing we did was see, can we pick up COVID with a smartwatch? And the answer is yes. Okay, here's the sign-up sheet, by the way. I'm trying to find my mouse. There it goes, right down here. Innovations.stanford.edu/wearables. So anyway, here's our very first patient. In purple is the diagnosis day, and red is the symptom day. And this is resting heart rate plotted for those who follow as a standard deviation plot. And wouldn't you know it, that this person's resting heart rate jumped up 9.5 days prior to symptom onset. You can't miss that signal. We wrote a real-time detection method I'll talk about in a minute. And so basically, this person's probably been spreading around his infection in advance of symptoms because he didn't know he was ill. And so we went on to show that basically 80% of people, you'll be able to detect this jump up in either at or before symptom onset in 80% of people. And the median is 4 days prior to symptom onset, 7 days prior to diagnosis. It's only 2 beats per minute, the stuff jumps up, but that's enough. When you're measuring somebody 24/7, it's very, very easy to spot that 2 beats per minute. So then we went on, and again, want you to all sign up here. We set up a real-time alerting system. And now we have 3 algorithms. This one's the best because it seems to be, we've set it, made it more optimized to be more efficient. But most importantly, it's a lighter weight algorithm. We're running this on a cloud-based system. We're following everybody on the cloud. And then if they get an anomalous increase, it's mostly increased heart rate, steps and other things come in. We're not getting all the raw data. That's the advantage. So this is why we want to work with actually Project Baseline, bring in more raw data. But anyway, if you get a shift up, you have to click on the app every day. Soon we'll ping you, but basically you get an information whether it's a red day, meaning there's an anomalous signal, or a green day, meaning you're at your normal baseline. And it works well, same number, 80% of the time, we can tell if somebody's getting ill at or before symptoms. Here's one of our first cases. 3 days prior to symptom onset, they were diagnosed the next day. 3 days, we could start getting these red alerts. But so again, it works well. It even works in asymptomatic cases. 14 of 18 asymptomatic cases get picked up. I forgot to say it works for both Fitbit and Apple. I'm sure it'll work for all devices because it's just the way it's built. It's looking for the shift up. And so here's a person who was asymptomatic. There's their diagnosis day, and they had red alerts running for about 2 weeks before that. So it actually, I want to emphasize it's not just COVID that triggers these alerts. Workplace stress is number one. Now we're running studies asking people to annotate this stuff. We'd ask you to if you do it, meaning annotate what your red alerts are so we know what's triggering it, so we can improve our algorithms. Intense exercise, like running a marathon, your heart rate stays up a few days, that'll do it. Alcohol intake, not just 1 or 2 drinks for dinner, but if you really tie one on, your heart rate will be up and it'll trigger these red alerts and so on. And other infections, of course, so we even picked up a mycoplasma one, which is kind of interesting. All right, so I can tell you it also worked for me on a personal level. That's shown here. It's kind of a fun story. So I did have a particular family stress event, mostly green alerts, kind of raw data showing you. Mostly green alerts, but there was a particular family stress situation. I had to fly out to see my daughter, came back, that can trigger red alerts, especially this one. And then I'm getting ready to go to New York City. This is a cool story. And I wake up and I'm a little bit congested. All right, is it allergies? What's going on? I'm not sure. So I do a COVID test and I'm negative, no sign of COVID. So then I check my app, and sure enough, I'm positive. So what do? I get on the plane because I figure, oh, this thing's negative. Maybe this is something else. It's not really COVID. Next day before you can go to the meeting, you got to get tested again. Bam, right, positive. And son of a gun, then I had to quarantine the whole week in New York City in this tiny little hotel room, all because I listened to my antigen test and not my smartwatch. So the upshot is that your smartwatch is more sensitive. It's less specific. It didn't tell me it was COVID, but it did tell me something was up. And had I listened to my smartwatch and not my COVID test, I would have been hanging out in California, which would have been 100 times more pleasant than this tiny little hotel room in New York. So that's my COVID story. I can tell we can also measure long COVID if you have long COVID data. So we want to become part of get data from Project Baseline. I believe we've asked for it either directly. I need to double check or through Joyce Leal here, who we're collaborating with on some watch stuff. And so we're very interested in seeing who got COVID and who has long COVID signals, which we can tell from smartwatches and maximum signals. So we have a study going on like that. I think this is just the tip of the iceberg. We've done machine learning to be able to try to see if we can pick up a signal on clinical things. And on the right are things, or on the left are things involved in anemia. So here's hematocrit, red blood cell, hemoglobin. See how we're doing time-wise. Oh, we're okay. These are all giving signals. They're not clinical grade signals, but they're good enough to see if something's shifting. and go get tested if something's off. Even fasting glucose and hemoglobin A1C can give a signal from a smartwatch, which we can, again, pick up. The other wearable I'll tell you about, we're doing all kinds of wearables now, is glucose monitors. We think these are super, super powerful. And I know Google got very interested in this with the contact stuff. Sure, they have other things going on. So we've been using, we got involved again, just to measure people for fun. We were measuring, at the time, these are being used for insulin-dependent type 1 and type 2 diabetics, but only the insulin-dependent ones. And so we wanted to see, well, what do normal people look like? So we started throwing these on normal and moderate pre-diabetics, I should say, and a couple of diabetics. And what we discovered is that even, so there's some people that have really good glucose control, that's fine. Some people are moderate spikers in their glucose levels, and other people are severe spikers, some of whom are just 100% normal by hemoglobin A1C and other measurements. I think it has to do with how the glucose dysregulation is measured. So we think these can be powerful diagnostics as well. And anyway, then we classified people. We wrote algorithms, came up with what we call glucotypes. So here's low glucose levels, here's moderate, here's severe spikers, and so on. So we can define these glucotypes. And then one thing we discovered, as did Aaron Siegel's group independently, is that different people spike to different foods, back to this personalized concept again. And some people spike to bananas, others to bread, some to pasta. Different people spike to different things. Nearly everybody spikes to rice. And this is work from Aaron's group, which basically you can see here's a participant, he doesn't spike to cookies. Now that's really unusual, by the way, but he does spike to bananas. And here's a person here who spikes the cookies, which is normal, but doesn't spike to bananas. So again, different people spike to different foods. We found the same thing. Some people spike to bread and peanut butter, others protein bars. Nearly 80% of people spike to cornflakes and milk. This stuff we think is like poison. So if you don't remember anything else from my talk, keep away from cornflakes and milk. That stuff will really spike you bad, and probably should be outlawed. Okay. All right, we built a dashboard. I'm sure you guys have one too. I know one was getting built. I don't know the latest in this, but our dashboard brings in wearable data, brings in clinical data, brings in omics data, and then you can display it in some useful fashion, whatever resolution, like daily, weekly, monthly, what have you, and follow your patterns at a personal level. And so this is what we think the future, good, I'm wrapping up right on time, should be. This is Mike Snyder's future. I'm a believer people get their genome sequenced before they're born. And then in conjunction with biochemical and wearable data, we'll be able to better manage people's health, predict, disease risk, catch disease early, monitor and treat disease based on personal data. I have an amazing set of collaborators, amazing set of people in my lab. Verily and Google keep stealing them. Tracy McLaughlin's our clinical collaborator, George Weinstock, our microbiome. These folks built the personal health dashboard. That's the genomics one I didn't talk about. Now we have a very large wearables team, all very focused on disease detection with smartwatches and other devices and CGM. So it's just a very fun and powerful group. And I always get asked about studies. I'll leave this up for a moment, but the bottom line is, so we have a lot of research studies. Just visit our lab website. You can probably get there through here. That's where the wearables one. And then some of my companies are running, letting you do profiling as well. And QBio is pretty related to this. January AI definitely follows continuous glucose monitoring. And we have a brand new one called YOLO that's actually doing a metabolic profile, which is kind of cool. So love to have you sign up for all these. So that's my story. And I should have saved, as I understood your timing, we have time for, questions, I believe, right, Ken?
Yeah, Mike, we do. That. First of all, thank you so much. What an extraordinary journey through your work and your personal history, which really sets this tone nicely, and I think will resonate very well with a lot of the Project Baseline participants, because they've had similar experiences where we've found things that are abnormal in their comprehensive assessments. And Some have been incidental findings that were nothing, but some have led to major procedures and important diagnoses. So a lot of what you said resonated with me, and I suspect many of the people on the call tonight. We've got now over 70 people participating, so it's terrific to see that. There's a series of questions in the Q&A, and I was gonna fire them off, and maybe we can keep the answers fairly brief so we can get through as many as we can.
Okay.
One is that, do you find wearing the devices makes people hypervigilant, or does it increase anxiety? Where's the balance in terms of what you've observed in the people you've studied?
Yeah, so we asked people, sorry, I'm trying to copy these links. I'll put in the chat, then I can take this off and see faces. So the answer is we actually surveyed people about that. And the answer is most people, now obviously it's a select group, but they found the information helpful and said it helped them manage their health better. So they use the information in a positive fashion. And every physician asks, oh, you're going to turn people into hypochondriacs, Mike. It's really a bad thing what you're doing. They used to say that about genome sequencing. They don't say it so much anymore.
Right.
So the answer is, I think, my own view is we always have a physician involved. So we're sorry, keep this brief, but we basically try and figure out from the patient, do you want to learn non-actionable stuff? Do you want to learn everything? The answer is yes. But some people say, no, give me only the actionable stuff. And so we just give them that. And so, and then most people are able to handle it because they've already set their expectations for what they want to know. And so we think that's the right way to do it. You should be in control of what information you want to get. I'm a big believer in that.
I think that's well said, and I agree. I mean, we were so worried that, or we were anxious that we were going to create the worried well with all of these digital technologies. And I think that with responsible sharing of information and engagement with participants in research and patients in the clinical care setting, you can avoid that. One specific question, have you done any work and gained any insights into white blood cell counts, both the prescribed range that we see on the lab reports versus an otherwise kind of redefined healthy range?
Yeah, well, people are all over the map on this stuff, actually, you may have noticed. So I wouldn't say we have it totally figured out, but there's no question we've detected shifts in people that turned out to be very, very powerful, especially people having cancer. It's every time you have infectious disease, you go off. It's kind of interesting. And people who get diabetes, actually, there's a very specific set of cytokines that go as well. So we're seeing different things. We may have a little bit of background discussion going on somewhere. I'm not sure where that's coming from, but anyway, yeah. So the bottom line is, I don't think we have that fully sorted out, but that's one of our favorite markers for looking for shifts in health is your cytokines and your inflammation markers. We have redundancy built in. You get it from both the metabolomics and from the cytokine profiles.
So someone put this question in as you were talking about food, but then I think you answered some of it when you were talking about individual responses to specific foods. And so if food is a medicine, how important is a vegan diet, or is it Mediterranean just as good? Or is it really individualized and some people are fine with meat depending on their own profile?
Well, I don't know the answer to that, but I'll bet the latter is the most correct. And the group as a whole, you may have seen this, they've now had a bake-off between Mediterranean diets and the keto diet. There's a study that came out on that's arguing the Mediterranean one is better. I'd like to see a few more studies because these things can be very population specific. And so I think we want to see how that's done and how that's trained. But I think at the end of the day, the diet that's going to be best for you, Ken, is going to depend on you. I think because it's going to come down to your microbiome. You lose a lot of your microbiome diversity as time goes on. And so I think maybe we need to boost it up a little bit with certain probiotics. And some of you may know I don't have any stock in this one, so I can say it. Pendulum has a microbe, Akkermansia, that actually drops your glucose by 1%, just a probiotic, which is pretty cool. And I don't know who that works well and who it doesn't. I do want to put the studies in the chat, but it doesn't go out to the group. So I'm not sure how to do that, Ken. And maybe you can just email it to people. I'll send it to you or something.
Yeah, if you send it to the panelists, I think we can get Maxwell or others to put it out to the broader group.
All right, I put it in the chat, so.
Okay.
Good, but you can't.
Great.
Anyway.
So someone's asking, I don't know how much you could share, Mike, but
Yeah.
you showed, I just have an Apple Apple. Someone asked, what wearables are you wearing right now? Can you share?
Well, here's Fitbit, sure. Apple, Garmin, those are the leading ones. And of course, my company, Sensomics, has one too. And why do we get it? Because it's hard to get the raw data. Most don't share. I hope Baseline's sharing the raw data, or at least, pretty early data. It's very hard to get that from Fitbit, and especially Apple. They're not very good at sharing. So that's why we had to make our own company. We're mostly using it for neurodevelopmental diseases, and we've formed a company around that sort of stuff. So, yeah. So
I think there's so many parallels towards between what you're doing and what we're doing in the Project 100%. Baseline Health Study. It's really terrific. One of the things that we're looking at also is a lot of the socioeconomic factors, including family dynamics and relationships. Have you integrated kind of relationship parameters and how important different types of family, social relationships and communities are? The question is specifically around, is it more important in longevity or in fitness? Love to hear your thoughts, was the question, the person asking the question.
Yeah, we're underpowered for that, to be honest. We do have people from different backgrounds, but there just aren't enough of them. And right. Same with, male versus female, we're just underpowered. We see a few signatures. I can tell you, though, we do have some families in the study, and that lets us tell the difference between environmental and some of the genetics we see. So on certain things, that'll tease out the environmental effects. We'll see, members of the same household are more similar in certain things, like certain kinds of your microbiome, not all types, but certain microbiome will be closer amongst family members than between families. So those are clearly environmental impacts. And I think this is where Project Baseline with this much larger cohort will be much, much more powerful.
Right.
Yeah.
Someone was asking specifically about at-home urine tests. Have you included those in terms of your collection of different parameters with the wearables?
Yeah, only modestly, in the sense that we've been incorporating, not for urine, but for the hemoglobin A1C. We have some at-home tests. I'm not sure what ones Baseline's doing. So we have a few that we run periodically as part, say, of the fiber study or some of the others. But we're very keen as a research project, I can't say a whole lot, but of doing home testing. And I'm pretty sure Verily is doing that as well. So anyway, it's a very popular theme, and a lot of people are reaching out to us these days about their favorite at-home tests. So we'll see how this plays out. I didn't get into this, but medicine of the future will certainly be home testing. You wake up and do your little prick and get, a biochemical profile. That will be a nice health monitor. I've always hated the idea of going to the doctor to get measured, because, you're definitely putting yourself at risk for getting ill when you do that.
So I really thank everybody for submitting questions. I know there's a few outstanding, but your time is valuable, and we want to wrap things up to let people get to family events this evening and dinner or other activities. So first of all, I want to thank again you, Michael, for spending the evening with us and for a really inspiring talk and sharing with you some of the really wonderful insights you've made, and reminding the Baseline community that's attending of really some of your pivotal role in the early days of trying to understand this effort, and some of the parallel things that we've done over time, and really some great opportunities we have moving forward. I also want to thank the Stanford staff. What an incredible group of people that work tirelessly every day seeing the participants, following up with the participants, and really helping to grow this wonderful community that we've been able to achieve over the last 4 years. And then finally, as always, thank you. Thank you to all the participants for the time, the effort, the commitment, and the enthusiasm to make this Project Baseline Health Study the success that it is so far, and the tremendous opportunity we have in the coming years to learn even more. So thanks again to all, and appreciate your time this evening. Take care, and we'll see you again soon. Thank you.
Yeah, by the way, I put my email in the answer. So one of the answers, that was my fast way. Any of you can reach out to me and send those questions, and I'm happy to respond. So
Well, that's incredibly gracious. Thank you so much.
all right.
Take care, everyone.
Great to see you all. Bye.